Hello everyone, my name is Beatriz Estevam, and this work is about sequence logos of SARS-CoV-2 Polyprotein 1AB cleavage sites, done at the Laboratory of Computational Evolutionary and Systems Biology at SENA USP. Unlike other virus in the beta coronavirus family, SARS-CoV-2 contains a large polyprotein coded by ORF1AB that is cleavaged by PAD-Pro and M-Pro, respectively papain-like and main proteolytic enzymes. As presented in the second image, PAD-Pro cleaves at the first 3 sites, and M-Pro cleaves at the remaining sites. Usually, these cleavage sites are well conserved because if they change drastically, the proteases won't be able to recognize them, impeding the correct production of proteins, affecting the virus replication and disease progression. This work aimed to identify the sequence and the variation of these cleavage sites. First, we downloaded completed SARS-CoV-2 genomes from the GSAID database and passed this data through a filter to maintain the sequences larger than 29 kilobase pairs and in which the proportion of uncertain nucleotides was less than 1%. Then we transferred the annotation to the genome sequences using the HAT software, so we used the feature match peptide as coordinates to identify the bordering regions to extract the cleavage sites. Then we translated the sequences using Embo's transect tool. To construct the sequence logos, first we compute the frequency of each amino acid and each codon in each position of the cleavage region, generating frequency matrices. Those were transformed into bit matrices using the function transform matrix from Python library LogoMaker. Then we developed a Python script to generate the amino acid sequence logos using LogoMaker. This library does not have an option to construct with codons, so we use the glyph class that draws characters on the graph at a definite position to construct the codon sequence logo. So the script defines the position of each codon to generate the codon sequence logo. Finally, we separated genomes based on submission date, determined using the metadata from GSAID, and using the same pipeline described before to generate the sequence logos by 7 days periods. We generate graphs with redundant and non-redundant data at the amino acid and nucleotide levels, and they are all available on Project's GitHub. The relations between the nucleotides and amino acid number of variants indicate that the sites between NSP9 and NSP10 and between NSP14 and NSP15 are highly tolerant to synonymous substitutions, since that the difference between the nucleotide and amino acid variants is close to 1, suggesting that they are under significant evolutionary pressure. Different than the situation on site between NSP12 and NSP13 that seem to accept more the synonymous substitutions, indicating a negative selection. The sequence logos based on redundant data show the most conserved cleavage site sequence, presented here on this image and on this table. In this table, we highlighted in purple the most conserved amino acids, but the variations on the sites exist and can be visualized on the sequence logos based on non-redundant data, where we can notice that the variability exists even on the most conserved amino acids. For example, for PAD-Pro, the glycine on the end of the proteins was substituted by a glutamine, and for M-Pro, the glutamine on the end of the protein was substituted by a histidine. Then we grouped the graphs generated by periods in GIFs, as presented here for bordering region between NSP6 and NSP7. Then we noticed that the number of variants grows around September to December, as presented on this table, coinciding with the alpha and delta variants' appearance and expansion. We conclude that the cleavage sites are well conserved, but variants exist even on the most conserved amino acids, and that the cleavage sites are under different selective pressures, producing a different number of variants between sites for amino acids and nucleotides. Thank you for your attention.